Okay. So, genome in 3D now. A little warm-up problem. Warm-up problem is actually, somebody already asked me about it. So, timescales. We've got some ideas about timescales. So what should happen during this timescales? DNA needs to be copied for the cell cycle to proceed. That's required. Now the question is there enough time to copy the DNA? Okay, so you know the length of DNA, and I can tell you how fast a cell can copy its DNA. And you can tell me how can it all work. So the number at which the rate of DNA synthesis, I'm using this fancy chalkboard, maybe I should. It makes my writing smaller.
Yeah, you're supposed to write big.
Yeah,
Leo
so I'll
says write big.
Okay.
If you want your lectures to last beyond this lecture, then
Maybe I don't.
that's
Maybe
why
that's
you,
another improv exercise. Rate of DNA synthesis. And synthesis is what stands for S in the S side, in the cell cycle, right? So that's what Simon reminded me. So G1 stands for growth phase. So there is growth, synthesis, growth, and then mitosis, right? You remember. That's what we do. So this S stands for synthesis. So synthesis. This rate is about 0.21 kb kb per minute. How long will it take for the cell to copy its DNA? And now think about bacteria and eukaryotes. Just check the number. You can use the, it's, okay, correction. It's base pairs per second. So it's slightly, so 1,000, okay, so I must have smoked something wrong in the bread. What do you mix in the coffee? KB per second, right? So bacteria, you know the length of the genome in base pairs. And so bacteria and eukaryotes, eukaryotes. How long would it take and how can cells accomplish this task? In the case of bacteria, timescales, in about 20 minutes. In the case of eukaryotes, a couple of hours for the synthesis. But in the limit, you can do it in 10 minutes. And that's a good exercise because it kind of gives you an idea of how things are, molecular machines operate. That's crazy. That's a good answer. I like it. Any? Yes, you had it.
So I was wondering, what does it actually mean? Does it mean like one that is moving?
Okay, so how does, okay, the DNA is being replicated, okay, that's a good question, using what's called DNA replication fork. So it's actually a machinery that copies DNA, and it makes two copies out of one. There's like a bubble. So the newly synthesized one, that's a newly synthesized one. And it's like two machines moving in opposite. Think about it. It's an approximation. Think about it as two machines moving in the opposite direction and making a copy of the, it's called the replication bubble. So this is a new one. The site where it initiates, this is called the origin of replication. I'm trying not to use biological notation, try to avoid it as much as possible. So this is newly synthesized or newly replicated DNA.
Do we have to have only one origin of replication?
Okay, so that's the answer basically. So if there were one origin, if
10 to 6 seconds
for
to get the whole genome.
for the human. So for the human, okay, good. For the human, it takes 10 to the 6 seconds. For bacteria, remember, human on the left, bacteria on the right.
10
For
to the 3.
okay, 10 to the 3 seconds.
But actually, for bacteria, it's order of magnitude of what's going on.
Okay, that's good. So the answer is it cannot work this way. For bacteria, maybe it may work this way, but it's kind of order of magnitude what we need. If bacteria divides every hour, or every 20 minutes, it takes 20 minutes to, if you operate at the limit, 1 kb per second, then you're fine, then you're kind of in business. If you operate at the lower limit, then you need a few factor 5 more. You need 5, 10 to the 3 seconds. Still of the order of an hour, but you cannot quite make 20, so it's hard to make divide in 20 minutes, right? It kind of pushes the limit. For human, there is no way,
Years.
right? There is no way we can do this. How can it work?
I don't know.
There are 2 solutions that nature found. So how can it work from human? Multiple origins, right? So you can have many places where this bubble opens up and progresses from there. That's time. So if you have multiple origins, but now the question is how many origins do you need? You need to replicate in the order of an hour. So how many origins do you need? You need roughly of the order of 1,000 origins. You need this. So that's an absolute necessity. Without it, you cannot replicate DNA. Origins of replication. These are origins. These guys. Okay. In bacteria, this is kind of slow part of the range. Longer part would be 5, 10 to the 3 seconds. And that's way too long, and you need to be done in less than 20 minutes. And you say, okay, maybe multiple origins. The answer is there is one origin. This is no. There is one origin, only one origin of replication in bacteria. How can it work? You may know the answer, so I'm kind of want people who don't know the answer to come up with this. How can it work? So there is one origin. If you start doing in one origin, it takes you a couple of hours instead of 20 minutes.
Can we have like kind of like cascade of
Yes. What does it mean?
So we make a copy, we start from one end, and then each end is like new copy, and then we start like new copy.
So
copy
this
down
is
before,
exactly.
and
I didn't follow that. Could you say that?
Okay, I'll explain. So the idea is that you start the replication, you have this is a new copy. This machines are moving, they started at this origin, and they are moving in the opposite direction. You produce your daughter's DNA. The cell starts to produce DNA for its daughter.
You don't wait to finish.
But you don't wait to finish, and you fire up from 2 other origins.
Oh.
And you start producing DNA for your granddaughter. cool idea. Now your granddaughter will have some DNA. And your daughter, actually for the other daughter, and then your daughter can start producing DNA for its daughter, for your granddaughter indeed. And in principle you can nest even more. You don't need to nest much. A couple of nesting events would be enough. But that immediately tells you that in bacteria you should have nested replication. And that's an absolute necessity. If you have one origin and this speed of the enzyme, and you cannot make this enzyme any faster, evolution didn't manage. So that means that you need to have nested origins. Nested replication. The problem that you're going to be facing now in bacteria is that it's going to be a mess. Because you need to be carefully making sure that this DNA is not getting entangled. You need to pull this DNA to the left, this one to the right, what they're going to do with the. So that's the blue and the white one, and then you get the pink one. Okay, the pink one goes somewhere, and then the green one goes somewhere. So the bacteria needs to invest lots of resources in making sure DNA is organized. And we only now start understanding this. I'll be talking mostly about eukaryotic systems, like bacteria has its own can of worms, like how to manage its DNA. That was a warm-up exercise just to illustrate the notion that different species deal with their DNA differently, but the way DNA is folded may actually be quite important for their function. Okay, so there is one more. I can move this one up. Okay, cool. Done with that. Cool. So, yes.
So there are two different polymerases doing,
There are there are DNA polymerases that fire up from the origin.
like, so one goes to each side. And
One goes into each direction, and then there is a whole. There are lots of molecular details on how they do this. And so yes. But there are two molecular machines that move in the opposite directions. Yes?
how do you know that there is only one origin of replication?
In bacteria, this origin has a very specific sequence. You can cut it out, and then bacteria is totally unable to replicate its DNA. You can insert it in two places, and then it will start replicating in two places. You can move it into another place. You can do lots of things like this. So the origin of bacteria is very well defined. I actually don't know what's known about origin in mammals. I will read it before the next lecture. Because a few years ago, it wasn't even known what determines where the replication starts in human or mouse.
So, but we know this 10 to the 3.
We know there are lots of them. We know there are thousands of them. You can measure the number of them. If you stop replication early on, replication lasts for several hours. If you look very early on, you see what are the newly replicated DNA. Using DNA sequencing technology, I'll be talking about this actually now. So biology is a field that's driven by technology. It's driven by two things. By A, technology, B, ability to use various biological machines that we discover to do new things in the experiment. That's kind of different from physics. In physics, we learn a new principle, and we can't really we now know another type of, I don't know. We learn about gravitation, but we can't go into the lab, and now gravitation will make us solve other problems much faster. Right? So we just
does, no? I mean,
exactly. We just learn about this. In biology, it's different. So technology drives things. So in the last decade, the ability, or 2 decades, the ability to sequence DNA, read DNA. Sequence DNA means read DNA. Really changed the way we learn about biology. And many things that we learned as rules in textbooks turned out to be not quite right when we start reading DNA and learning about stuff. I'll be talking in a few minutes. I'll talk about genomic technologies, but the way how we can learn how many origins are there, we can add a modified nucleotide. So newly sequenced DNA will be different from the old one, chemically different. And then I can read only new DNA. So I can stop this cells in the middle of the S phase and then sequence all the newly synthesized DNA. And I will see that all the newly synthesized DNA only matches about 1,000 different places, actually more than 10,000 different places. So that's a low limit. We need more than 10 to the 3 origins. And the same I can do in the cube. So let's close this now. We have another problem now. The problem is that we need to pack very long genomes in very small cells, in very small nuclei. The first question we can ask, okay, say we have a meter of human DNA. How big would be this meter of DNA as a physical object? It's not going to be 1 meter long stick of DNA, right? So it's going to be kind of flexible. So how do I know how big is this piece of DNA? So the question is, if I let it do whatever it wants, how big is this thing? What's the radius of this thing? So to do this, I need to start talking about polymer physics, so that's going to be a little deviation to polymer physics. And again, I'm sure many of you have heard this in your classes, but so I'll just repeat this. So what's a polymer? A polymer is a long molecule, and there are many different classes of polymers chemically. I'm not going to draw chemical formulas. So some of them, for example, can consist of rigid bonds and different angles, and then you can you can have rotation around these bonds, and for example, fixed angles. So things can rotate around bonds. So that's going to be like a puzzle, like kids' toy, because this kind of rotating around bonds. You can have polymers that are continuous, like DNA. It does not have segments and joints. It has a continuous shape. The way to quantify this structure or that, if I zoom out of this one, if I look at many of such joints, it would also look kind of like a continuous chain. At some level, of course, gradient. And the way I can quantify this, I can ask, if I draw if I look at the direction of this segment at position S, and then if I look at the orientation of the segment at position T of S plus S plus L. So if I look basically at the angular, and then I can look at the angle between these two vectors. I can look at the angle between T of S and T of S plus L. Right, so that will tell me how much freedom do they have in terms of orientation. So for example, if this piece of DNA were like that, then these two angles would be basically the same, right? If this were like incredibly rigid piece. If it were very flexible, these angles would be all over the place. So I can look at an average angle, average over all positions S. So that's going to be some function of L. It will just depend on how far the 2 points are, and ask a question, if I have 2 points separated by some chain of length L, do they always point in the same direction, these monomers? Or are they totally free to do whatever they want? So it can be shown that this actually decays exponentially with L, and the decay length is called LP, or persistent length. I just realized that I'm not wearing the microphone. And I'm facing you this way, so that should be on the right side. Okay. Now you can hear me. You should be able to hear me better. Okay. So I'll just quickly repeat myself. So basically, I'm looking at the orientation of two monomers on the polymer. If the polymer is continuous, then I will just use tangential vectors. And then I will look at the angle between them as a function of what's called chemical distance. Distance between two monomers along the chain. This is called chemical distance. Because again, this is how many, okay, L. And I will average over all possible positions of this monomer to a pair of monomers along the chain while keeping their chemical distance constant. So they're always distance L apart. And then I ask what's the angle between these two things. And this angle in a polymer, in a freely jointed polymer, so this theory is called freely jointed chain, freely jointed chain. In the freely jointed chain, this angle is decaying exponentially with L, with the separation between them. And the characteristic decay length is called LP, or persistent length. So what persistent length characterizes? It characterizes the fluctuations between two monomers, but it's a length scale. So it basically tells you on what length scale these two elements remember each other. Or in other words, if I have a monomer like this, and I go along the chain of a polymer, I ask how far can I go such that I still remember this orientation. So over LP, I remember this orientation. Over much longer than LP, I don't. So that's the first result. Then the second, now you can ask the following question. If I have a polymer like that, and I'm interested in understanding how the end-to-end distance, R, depends on the length of the polymer. So now I want to answer a question of what's the end-to-end distance, this end-to-end distance, R of of L, capital L, where capital L is the total length of the polymer. Capital L, length of the polymer. So I want to know end-to-end distance as a function of the length, and I want it to be averaged over all possible conformations of this polymer. In this case, in equilibrium, there is no energy, there are no constraints. What did I neglect in doing this? Tell me, so why freely jointed chain is a good or a bad approximation of stuff? Because I want you to think, okay, so freely jointed chain basically tells me that the polymer can do whatever it wants as long as two neighboring things kind of are oriented in the same way. Yes? It can cross itself. So it has no topology. Okay, cross itself actually implies two things. One is excluded volume. Second is topology. Topology means that if I start with, imagine I'm dealing with a ring, and I explore all possible states of a closed ring. It can cross itself, and it can knot itself into all sorts of knotted structures. This is really hard to take into account. There is another consideration, is that it has a volume, right? In reality, polymers don't look like that. Real polymers have volumes. Okay, right here. It's a tube, right? And this tube bends somehow. So, what did I draw? Very complicated. Okay. So it's a tube of some shape, and it cannot cross itself, not only because of topological constraints, because it just excludes volume. This monomer and this monomer cannot come to each other too close because of the diameter of the tube. That I can take into account by approximating it by a series of monomers. But then in addition to what I'm writing, I need to then somehow take into account this excluded volume. And that we can do in one relatively easily. But so far, freely jointed chain, no excluded volume. So, okay. Note is that there is no excluded volume. And no topology. No topological constraints. In this case, I can write down the average end-to-end distance, and it will actually equal to, and I want it squared. So the squared average end-to-end distance will be 2 times L times LP. And I'm not going to derive this, so I'll just, we wouldn't have time for this. So that's an important result, because it basically tells me that the end-to-end distance depends only on the length of the polymer, and it goes as length to the power of 1/2, and on the persistent length. And moreover, I can even combine L and LP into a new quantity that I will call B, and I will simply define B as 2 persistent lengths, and this is called Coons length. Why is this a useful, why is this a meaningful thing? Again, as a side note, if I were to ask, so note, if I were to consider a random walk of monomers of length B, all of them supposed to be of the same length, B, and it's a random walk, so it's completely random, so this can go anywhere else, so on. Then the end-to-end distance of this random walk, R squared average, will be n, the number of such segments, times B. Times B squared, sorry, yes. I was like, something is wrong. Yes, now it's where n is the number of segments. This is true actually in any dimension. That's a nice thing. It's okay. So then I say, okay, basically it means that my polymer behaves like a random walk with segments that are twice the persistent length. So that means that I can describe my polymer as a random walk of sticks and the size of the stick is twice the persistent length. So I can approximate this by this things of size B. Great. So now we can use, so now if, okay. So the scaling here is useful because what we also notice is that generally R will scale as n to 1/2. This is freely jointed chain. If I were to add excluded volume, and I need a little bit more space, let me think. I will start using this later. If I were to include excluded volume, now with excluded volume, I can, so with exclude volume, polymers can be approximated not as random walks, but as random, self-avoiding random walks. A good model for a random walk. So this, okay, good. So I will use this diagram. So a good model for this polymer would be a chain on a lattice that cannot cross itself. So that would be a self-avoiding random walk. SW, self-avoiding random walk. And for the self-avoiding random walk, the scaling is different. For the self-avoiding random walk, R scales as approximately n to the power of 0.6, again close to 0.6. This number can be computed, 0.6 can be derived if I had another 15 minutes, but it's an approximation, and exactly it can be calculated just numerically.
And that is in 2D.
This is now in 3D. This now, okay, that's a good point. So this coefficient is universal. This one is not. This is only in 3D. Thank you for the note. This depends on the dimension. So that means that if I take into account the excluded volume, I can also do this calculation, but instead of having power of 0.5, I will have a power of 0.6. Okay, so that was a simple, so we'll come back to polymer physics again, but before doing this, we would love to get an idea to answer this question. So how big is this piece of DNA if just left alone? Maybe it would just fit inside a cell if it just jumbles the way it wants. So I want you to calculate R for the whole genome, where you take L of the whole genome, and also take R of one chromosome. In bacteria, there is only one. Okay. In many bacteria, there is only one chromosome. In mammals, in humans, how many chromosomes do we have, by the way? 23. Good. So they vary in length from hundreds of megabases to tens of megabases. So so for yeast, Baker's yeast has 12 chromosomes that vary in length from a fraction of a megabase to a megabase. So roughly, so for yeast there are 12 chromosomes. For human there are 23 chromosomes. So I want you to get an idea of how big is the whole genome and how big is one chromosome. Just pick a number. So it's guesstimation. And do it for bacteria, for yeast, and for humans. And so we'll continue this. And forget about the excluded volume for now. Let's say that the excluded volume can be neglected. What you should have asked me, but you're silent. What's the persistent length? Exactly. So LP for DNA has been measured quite accurately, and this is 50 nanometers or 150 base pairs. So that's a super useful number. And I will do a drawing, actually. Okay, any numbers so far? Go ahead, sorry.
36 square microns.
Okay, I want the distance, so it should be in linear microns. Size. So basically what we're trying to estimate is the size of a random jumble of DNA formed by the whole human genome or one chromosome of the human genome, and the same for bacteria. Does it fit the nucleus? Does it fit the nucleus here? Does it fit the nucleus there?
10 millimeters.
Okay, let's do everything in microns. So for the whole human genome, what did you get? Ballpark. 10,000, 10 to the 4 microns. So it's any other suggestions? Okay, give me numbers, one at a time. 500 for the whole genome or for one chromosome? For the whole genome. 500, okay, we get many numbers. Okay, any other numbers? Any numbers? I was once teaching at the PFL in Lausanne, and so the audience was silent. So I took a hostage. I took somebody who was sitting in the front row and I was asking personal questions like, okay, so what did you get? What was your number? And so that was my way of waking people up. I will have to take a hostage now. 10 to the? 10 to the 5. What units? Meters? Micrometers.
I would like to correct my...
Okay, correct?
So it's 300 micrometers.
300, okay. I think we're kind of converging, okay. So do you agree? I'm taking a hostage. Good. Okay. 300 nanometers, 500, microns. That's the whole genome. So would it fit into the nucleus? The nucleus is 10 microns. It wouldn't. So it's even if it's compactifies by itself, if just this is 300 microns. Okay, how about one chromosome? One chromosome is 20 times shorter, so the genome would be 20 times shorter. Because of the scaling of square root, that's going to be square root of 20, say 5. Right? So it's going to be 5 times smaller. So one chromosome, if this was, so that would be 60 microns. That's a long chromosome. A short chromosome would be 100 times smaller. So a short chromosome, 100 times shorter in length. So, more than that. Yeah. Okay. So that would be 10 times smaller. So this can be as small as 30 to 60 microns. It wouldn't fit into a normal nucleus, but it's kind of borderline-ish, right? Maybe some nuclei are big. Okay. Yeast and bacteria. And in yeast also get an estimate for one chromosome. This was 3 10 to the 9 base pairs. This was 10 to the 7 base pairs. That's 5 10 to the 6 base pairs. So this is 300 times shorter, right, in length. So take a square root of 300, you get 15. Whatever. 17. So this should be 15 times smaller. So this is like 20 microns. Now I need to divide by 12 chromosomes. So if I would divide the length by 12, the size would go by square root of 12 by, say, 3 or 4. So this is like 5 microns. Okay, so that's interesting, because now it looks like one chromosome of yeast, individual chromosomes, can fit inside the nucleus. Okay, and then finally bacteria. This is 1,000 times shorter than the human genome, so it's 30 times smaller in length. So what should be the number? Yeah, something like 10 to 15 microns, right? One chromosome. So that certainly would not fit. So now we have two kinds of situations. This is not going to fit. This is most likely not going to fit. So does not fit. So they do not fit as a random object. So this is 300 microns, and the size of the nucleus is 30 times smaller, something like smaller than this. This is, so it does not fit there. The whole yeast genome as one molecule would not fit either, but an individual yeast chromosome can fit in. So that means that if I have a yeast nucleus, it's a budding yeast, so it's a little bud, then one chromosome may actually be able to spread around. So I don't really need to compact individual chromosomes in yeast. They just fit. More or less, maybe some of them, shorter ones, longer would have to be compacted. Okay, and then bacteria clearly does not fit, because bacteria is much smaller than that, an order of magnitude. Okay, so in doing so, so that's an important, so that's a more kind of, just to get an idea where we stand. So it tells us DNA needs to be compacted in some way. In doing so, we continued using this approximation. We disregarded the excluded volume. In reality, there is excluded volume of DNA, which would make DNA swell. It would make it worse, exactly. But the effect may not be so big, because DNA by itself, that's why I drew DNA here, is actually quite thin compared to its persistent length. Persistent length is 50 nanometers, 150 base, and the diameter is only 2 nanometers. So it's a relatively thin, flexible polymer. So maybe exclude volume is not such a big deal. Again, you can say electrostatics would make things repel each other. So exclude volume may start playing some role, but maybe not such a huge effect. And it would make things worse. Okay, so these are the estimates. So with this, let's go and take a look at how nature solves this problem. So now we know that nature needs to solve this problem. So the warm-up exercise was to show you that when you put numbers together, you see that nature should find a solution. That it doesn't work the way it cannot just work the way it is by itself. There should be a mechanism. So here there should be a mechanism. And now, so we're talking about genome in 3D. So we're continuing with genome in 3D, and now we'll start talking about chromatin. So what's chromatin? Chromatin is a way. Chromatin is technically defined as DNA plus proteins bound to DNA. DNA bound proteins. So nature solves this problem by packaging DNA by putting some proteins that locally compact DNA.
So this here was that the polymer is kind of in free space. So in principle, I mean, it could also be that the boundary is just.
That's a great question. So far, we consider it an unconfined polymer. So the polymer can be confined. You can just put polymer inside a small cavity, and it will fit whether it wants it or not. But that's the point. So is this? So that's going to be our next model. We'll come to this in a minute. So let's yes.
And do you consider everything like volume-wise? How much, like what is the percentage of the
Ah, yes. Thank you for reminding me this. That's also what I wanted you to compute. What's the volume fraction of DNA? Yes. And I had it in my notes. So, good. Let's do this last. Oh, there is another. No, I already used the space. Let's do another estimate here. What's the volume fraction of DNA? That's a super useful thing because people always assume it's a very different thing. Volume fraction of DNA. Here you don't need to do it by chromosome. The whole genome. Yes, thank you. Oof. I need to start looking at my notes. Volume fraction. Before we go into volume fraction of DNA. Okay, so to answer this question, you may want to ask me, so how big are the nuclei? We kind of already did this somewhere. Over there, yes. So okay, let's say it's 10 by 10 by 5. Approximate. Let's say 5. Let's say the radius 4 microns. I don't know. Maybe even more permissive, 5 microns, such that it fits. 5 microns in diameter, and you don't even need. And that's relatively spherical. This one is like flat-ish. You can completely forget about like nucleus being round because for two things, we don't know these numbers exactly, and second, so it varies from cell to cell. You can assume that the nucleus is like square or like cube. Okay, what's the volume fraction? Make an estimate. That's a very useful number. Diameter of DNA, that's why I drew this, is 2 nanometers. I will start drawing here. Okay, in
Yeah, so like,
for human,
yeah, if it were a square, like it's order of magnitude of like 20%.
20%, yes. Any other numbers? Approximately 20%. So that would depend on the way you calculate it.
For yeast is like 0.4%?
0.4. Where, in yeast or where? 0.4? 0.4%, okay, I'm surprised. Okay, maybe, okay, I'm sure there are going to be more numbers because there are more people, so, but everybody's silent. So the volume of this, like of the 100 cubic nanometers. Here the volume is like 500 cubic microns. Okay, 1%, okay. I think we'll have to vote. Okay, so that's the range. Check 1%. Any other estimates for human? For the whole genome, not one chromosome. You got 1%? So what did you get for the volume of DNA? Three squared nanometers, right, of the cross section. It's one. That's the cross section times the length. The length we have already in micrometers. So this is 3 nanometers, 3 10 to the minus 3 if we want in microns, squared, 6 nanometers squared times 10 to the 6 nanometers. So what do we get? 3,000. Oh, we get 3. Sorry, it's all microns, right? 3 micron cube, exactly. The total volume of the cell is, of the nucleus is like 500, so it's of the order of 1%, so that's not 20%. Huh?
So it means that we don't use 2 and 4 in genome.
Okay, if you used, okay, good. So if you use 2 and 4, that's a good correction. If you use 4, that would give us like 4%, 5%. Let's round things to 1s or 5s. Okay, 1 to 5%. Because it's a cell. Who knows what the cell is? Maybe it's a tetraploid cell. Maybe it's a G2 cell. Then it's 4. That's reasonable. Okay. In yeast, you got 0.4, half a.
Divide by 25. So like 0.016.
Point.
No, 0.16%.
0.16%.
No, it is 0 after the point.
Oh, there is 0. Okay, that little. Any correction to that? 0.2%. Okay, that's looks better to me. Okay, so this is again 3 10 to the minus 6 micron squared of diameter times 6 10 to the 3 micron. This gives us 20 times 10 to the minus 3 micron cube, or
0.02. 1, 2, 3. So 0.02 micron cube. And the typical volume is about 100 microns cube. That's weird. Did we get the numbers right to begin with? $20 micron. Okay. Okay. So that's indeed closer to that. Pi times r squared. R is 1. Okay, so we get somewhere in the fraction of a percent. Bacteria. About 6%. 0.6%, yes. So tiny. So the take-home number is that we're talking about relatively small fraction volumes, even in human, but we're kind of, somewhere in the fraction, it's not negligible. But generally, it's not a problem of fitting DNA inside this volume. The volume is largely empty.
How are you going to fit it?
Yes, it's how you're going to fit it. And the first level of fitting the DNA is wrapping DNA around little barrels called, the barrels are made out of a protein called, proteins of that generally called histones. So this 8 histones come together, form a barrel. The barrel is wrapped up, so DNA wraps around the barrel, and you have these barrels every 150 base pairs of DNA. You have one barrel. I will draw this. So you have DNA, it wraps around the barrel, goes to the next one, wraps around the barrel, keeps doing this. So these are these barrels. Size of the barrel is such that with DNA, it's about 10 nanometers in diameter, 6 nanometers in height. Each barrel takes about 150 base pairs of DNA, 147 on average, with some linker. So that's this linker. The linker depends on the organism, and we can say 50 base pairs, roughly. That's the linker, and then within the barrel, you have another 150 base pairs. So how much linear compaction does it, so you wrap, you go, you wrap, you go. How much linear compaction does it provide you?
The order of twice, or like an order of 1 multiple of
Well,
1.
you trusted me too much because you trusted that I made 2 turns. I didn't tell you how many turns does it make. So I need to tell you how many turns it makes, and it makes 1.7 turns. 1.7 turns. I wasn't sure how to draw it. So I said, okay, 1.7 turns. So
with physicists, 1.7 is equal to 1.
Yes, practically. It's 1.7 is closer to 2 for me, but it depends on what you count. If you count your salary, then it's closer.
It's closer to 1 than to 10.
Yeah, with French taxes, it's closer to 1. With American summer salary, it's close to 2. Right, but so that's right. So it's roughly pi times the number of turns. So the shortening is number of turns times pi. Actually, I have a funny story about this. So about 7 years ago, in late February, I get an email from a journalist. And she wrote to me that I'm a journalist working for CNN Science. I said, oh, I'm honored to get this. And I'm preparing an article. I'm like, yeah, that's interesting. Okay, maybe since she wrote to me, maybe something about me. And I prepared an article for the Pi Day, March 14th, 3/14 in the American system. Do you use number pi in your research? Shit. And I really want to be on CNN Science, but so do I use number pi in my research? Like, I said, okay, thank you, I'll think about it. And I was like, okay. And I was like, for a couple of days, I was just thinking about this in the background. And I said, yeah, that's why I use number pi in my research. It's number of turns of DNA times pi that compacts. That's how much you compact DNA. So I wrote a story about this. So it may still be Googleable right now if you look for Pi Day nucleosomes. And then say, can you make a picture for me? So, okay, that's how I got famous. So I figured out that I use pi in my research. So this gives us about 5-7-fold compaction. Linear compaction. Okay, so that also changes a little bit the game we've been playing, because now we're not dealing with DNA anymore, we're dealing with chromatin, which is a fiber of about 10 nanometers in diameter. So I will approximate this now as a fiber, right? Of 10 nanometers in diameter. And that may have a different volume in the nucleus. And I don't want to spend time actually computing the nuclear volume of chromatin, but we can do this later. And it shortens DNA. Will it solve our problem or not? As far as what was the challenge? Need to figure out how to. Okay. Will it help us fit all this DNA inside nuclei? We've got 5-fold compaction, linear compaction. How much will this change the size of the DNA if it's just a random coil of DNA? Square root of that, so it's like a factor of 2. So a factor of 2 is not going to make a huge difference here. A factor of 2 may help there because 30 will become 15, and the human nucleus is like 10 microns in cross-section, but maybe in bigger nuclei, some short chromosomes can actually give me, can fit as random coils. So that's actually a terminology that I should have introduced. So when we were doing this polymer physics, what we were talking is about a random coil. Random coil of the polymer. That's the state that's described by the freely jointed chain. So what we're saying, so that's the size of a random coil, because now we'll have to introduce other polymer terminology. So random coils are not going to may fit for small chromosomes. Random coils, again, for square root of 5 will make a huge difference for yeast, because now many chromosomes can fit. Here we were saying one chromosome. Sorry, that's too high even for me. We were saying that one chromosome of yeast can fit inside the yeast nucleus if this were naked DNA. It may fit here. And it's not going to help in bacteria. Well, first of all, in bacteria, there are no nucleosomes. Sorry, that's a major thing. So this is only in eukaryotes. Only super important. Strictly speaking, again, not quite correct. Because archaea, which is a different kingdom, has its own histone-like proteins. But we know so little about archaea and how it packs its DNA that it's kind of too early to discuss this. But certainly not in bacteria. So that may help yeast. You should have asked me one question. Yes? Yes, thank you. That's the question I wanted. So I assume that it does not change the persistent length, but maybe it does. What are your expectations about changes in the persistent length? Will it make it longer or shorter? Who said? Yeah. Why longer?
Slightly longer.
Why?
Because there would be more replication, maybe because the persistent length is also due to electrostatic.
So stuff would get kind of bulkier. So LP can get slightly longer. Any other opinions?
But if you take into account electrostatic interactions, then it should become shorter because now we have higher.
Well, let's assume that electrostatics is already taken care of because there are enough counter ions. We're talking about physiological conditions, sufficient salt. So it's all kind of in nice and sort of nice soup of stuff. So my analogy started kind of getting different closer to lunch. So it's all in this soup. Right, so it can get shorter. So let's estimate LP for this file. Yeah.
You could also just introduce extra twist and make it not really shorter or longer, but sort of make it polymers that actually wants to twist rather than wants to stay straight.
You can, okay, so that's a huge can of worms. It's a Pandora's box. Supercoiling. You can do lots of things to polymers by twisting them. I don't want to talk about supercoiling now. It can change. It can make polymer that's naturally twisted or naturally has some shape. Because I didn't tell you how these nucleosomes are arranged. In principle, they can stick to each other and form some sort of a structure by itself. So far, we assume that they are not doing this. And so far, there were no serious evidence that they do that. The evidence might be emerging now, but so far it looked like nucleosomes are just beads on a string. But it's a good comment. We should talk about supercoiling maybe tomorrow. Okay, let's estimate the persistent lengths. Let's assume that these pieces of DNA, the linkers, are as flexible as they were before. If there were no excluded volume, what would be the persistent length of this polymer? As long as I accumulate enough persistent flexibility of these linkers, so persistent length of DNA was 150 bases, so I need 3 such linkers, and that would be as flexible as the persistent length of DNA. But now, these 3 linkers are forming 3 nucleosomes, each 10 nanometers in size. So this is like 30 nanometers. So we're roughly still the same, like 50 nanometers before, but it got shorter, actually. But because they repel each other, maybe it's not 30 nanometers, maybe it's 50 nanometers, maybe it's 100 nanometers. Ballpark, we're staying the same. Maybe a factor of 2 in the persistent lengths. The surprising thing is we don't know the persistent lengths of this fiber. It's very tricky to measure, and so far nobody succeeded in estimating this number. But we can say to the first approximation, this fiber persistent length is somewhere of the order of maybe 3 to 5 nucleosomes. So this is somewhere, again, nucleosome plus a linker, maybe 50 to of the order of maybe 50 nanometers, somewhere, ballpark. That's not that different. So we can say that maybe persistent lengths didn't change much. It got bulkier, but it compacted DNA linearly. So it may help yeast. It's not going to solve our problems for human cells. Maybe if very short chromosomes and very large nuclei. Okay. Now beyond that, beyond this beads, so the take-home message from this is that now we're dealing with a beads on a string, nucleosomes, beads on a string polymer of 10 nanometers in diameter. So that's this polymer. And this is called 10 nanometer fiber. So we have now 10 nanometer fiber of DNA that we need to compact inside the nuclei. Now sort of we're at the point so that from this point on, it's not really known how chromosomes look like inside the nuclei. So how this fiber is formed inside chromosomes. You can look inside and look at the whole chromosome. We have 15 minutes. Okay. If you look inside cells and ask, so that's at the scale of 10 nanometers. How do chromosomes look like as a whole? Like, can I do microscopy? Why are we guessing? Why are we not doing microscopy? So if you look inside a nucleus of a human cell or a mouse cell, you can visualize individual chromosomes. And there are a couple of interesting observations. One is that individual chromosomes occupy some volumes. So if this was a chromosome, I would say this is chromosome 1, maternal, like what we discussed earlier. And then you would have chromosome 1, paternal. So they occupy some volume inside the nucleus. And that's already interesting and surprising. So, and now we're talking about interface. Collectively G1 as G2. So each chromosome does not occupy the whole nuclear volume, though as a random coil, it should be 10 times bigger than the whole nuclear volume. So that means that chromosomes are organized and compacted quite a bit. Now, if we now go into the M phase, M stands for metaphase or mitosis. Now, if we go into this phase, then chromosomes start looking like elongated bodies that I'm sure you've seen many times. So individual chromosomes separate in space and become elongated. And at this point in the cell cycle, usually there is no nucleus. Nucleus breaks down. Chromosomes are just aligned and spread like that. There are more of them. Typical lengths, again, we're talking about a few microns, 1 to 5 microns in length. And the diameter of a chromosome would be something like 0.6 micron, half a micron. And again, the cell cycles between this state and that state. So we really don't know how this fiber is folded inside this body. And this body is few microns in cross-section. Before, yes?
I'm wondering, when you draw it like this, is it all the mother chromosomes are in the blue, or is it just one chromosome?
I'm drawing one chromosome 1. So I can draw, so another chromosome 2 would be, no, so maternal and paternal are not segregated. And not aligned in, they're aligned in flies. And they're not aligned in other studied organisms. But they may be aligned somewhere else.
So here chromosome 2 is somewhere else in the, like...
Yeah, chromosome 2 would be somewhere else, yeah. So 2 would be here, 3 would be somewhere else. So they're completely randomly assembled. Next lecture I'll show more slides. I don't want to take time and sort of turn on the projector. But this arrangement of chromosomes is completely random. With some exceptions of some chromosomes having a tendency to be in the center and some having a tendency to be the periphery. To the first approximation, it's random.
Random but segregated.
Yes, random but segregated. So chromosomes have their own territories. So this is called chromosome territory. So kind of a region occupied by a chromosome is called chromosomal territory. So they're segregated. Now we can go back to the very beginning of the lecture. And so remember how the genome is, 1D genome is organized. It has genes, and then gene-poor regions, and then gene-rich regions, and then gene-poor regions, and so on. I'm waiting for a question. So that's how every chromosome is organized. You should ask me something. How are poor regions, or where are gene-poor regions versus gene-rich regions in space, right? So they are actually, so there is some level of organization of this. I need different color for them. So if I look again at the interface, and now I ask another question. So chromosomes are not organized in any particular fashion. Moreover, generally, why genome is broken into chromosomes is not entirely obvious. For example, in yeast, some species have as many as 16 chromosomes, and some have as few as 3. And they're fine. Moreover, recent experimental work from NYU show that you can take yeast and you can fuse its chromosomes together, and it's fine. Instead of 12 chromosomes, it would have 8, and then 4, and then a Chinese group managed to do even 2 chromosomes, and there is no obvious difference in the phenotype. 2 is kind of extreme, but with 4 instead of 12, fine, totally fine. So presumably you need to break genome into chromosomes to separate easily, but so far it wasn't obvious why you need to do this and where you cut stuff into chromosomes. So moreover, you can even have some interbreeding between species with different number of chromosomes. Like, yeah. Well, these are different species, so they interbreed only for one generation, but that's it. So you get non-productive kind of offsprings, but otherwise.
So you get an offspring?
No, well, you get one, yeah, you get.
With how many chromosomes?
Yeah, was a mixture,
What?
Yeah, like, well, yeah.
No.
So people sequenced actually, so that was an interesting, like this hybrids of donkeys and horses.
Yeah.
There are 2 different ones.
Yeah.
Depending on the male and the mule and the, what's the second one? Exactly. No, there is.
No, that's something else.
No. There is a mule and there is. Yeah, there is a name for it. I forgot. But there is zebra and.
Yeah, that's the
Yeah, and so they have different number of chromosomes, but they're fine, kind of for one generation.
So the new one has the lesser, or just something in between?
They should presumably have a combination of full set from the mother and full set from the father.
What?
How else? Kind of normal.
I don't know.
An oocyte should have a full set of chromosomes, and the sperm would have a full set of chromosomes. So.
Then you think you just have to throw a discard out.
But they remember, so you don't need to pair these chromosomes. It's just a collection of genes. So that's the whole point. You can cut genome into chromosomes differently, and that's okay. One idea was that that's how you prevent further. So then from this point on, that becomes problematic, because then you cannot segregate them evenly. You cannot make one complete set of genes out of that. Because then you take paternal number 1 from the horse and maternal number 1 from the donkey, but so they're not the same. They have different sets of genes. So one idea was that splitting into chromosomes would be.
A horse has 64 chromosomes and a donkey has 62. A mule inherits 63.
Exactly.
And the source
61.
Sandra posts in an article called "You Can't Fool Mother Nature."
Right? One hypothesis was that's the mechanism of speciation. And for yeast, it was shown that once you start merging chromosomes, yeast, again, Baker's yeast can be a sexual species and an asexual species. It can decide how it behaves. And so when you try to mate them back, you cannot mate them back if the number of chromosomes is vastly different. You can mate back, I think, 8 to 12 chromosomes, but you cannot make 4 to 12. So I don't know whether it got published or it was in bioRxiv, but the paper is out. Okay, chromosomes, not entirely obvious what role they play, but there is another level of organization of the genome into gene-rich and gene-poor regions. And that's highly organized. So generally in human and mouse, and gene-rich regions, and I was drawing gene-rich regions in magenta, right? So these were genes. And then there are going to be gene-poor regions in between, and I will draw them in green. So these are gene deserts. So gene deserts are typically at the periphery of the nucleus, and gene-rich regions are typically in the center of the nucleus. So this is highly organized. Organized in a sense of phase separated.
I don't quite understand. So supposedly this was a large strain, and we coiled it around a bunch of histones. And so how is this organization happening?
Okay, so histones can be abstracted out. Now it's a different type of polymer. It's not DNA, it's a fiber.
Yeah.
But this fiber consists of magenta regions and green regions. Magenta regions are full of genes, and then green regions have fewer genes. So that becomes what's called block copolymer.
Is this before we fold it around the histone or after?
After. Histones are already there. Histones are always there.
Okay.
We spoke about everything other than histones just to introduce the concepts. So histones are there. So it still consists of blue and magenta regions. Now it's folded in such a way that the green regions are at the periphery and the magenta ones are in the center. In other words, it's folded in such a way that sort of there's a little green one, a little green one, a little green one, and then magenta ones are somewhere coiling here. So that's a blow up of, say, this part.
How does the cell know?
Exactly. How does the cell know which part is which and where each part should go? So that we'll discuss next time. So I just want to already tell you this result. So that's the fact. That's the observation. Now, how can this? Yes. Okay. So let me rephrase your question. The question was whether histones are organized evenly or as densely in green versus magenta regions. Again, magenta meaning lots of genes, and green. At this level, at this scale, they organize similarly. But yes, if I zoom in, histones are organized somewhat differently at the beginning of the gene. The very start of the gene is missing one histone. And that I plan to talk about this, but given our pace, I decided to skip this part. In principle, yes, histones are not organized. We need to stop now. So histones are not covering stuff uniformly, and that there is a rich and beautiful physics in how nucleosomes are organized. I'm trying to zoom out at this level. So the question is, how is genome organized? So first of all, we know now that there is a fiber of histones. This fiber of histones is folded in such a way that active regions are in the center, inactive ones, no genes or few genes, are at the periphery. And the question is, how is it all folded? How is it all kind of jumbled inside? And that's what we're going to discuss next time. And I think I should stop here.